Right, so welcome everyone to Monai Day. So now we're gonna switch gears a little bit and talk about 2D images. So my name is Andriy, and I'm gonna talk about VISTA-2D project we contributed to Monai, and it's about cell segmentation. It would not be possible without our core development team, which also includes Vish and Dagwan. So VISTA-2D is a cell segmentation training pipeline in Monai, emphasis on training. So with this, you will be able to segment cell images, which are microscopy images, which look like this, and get this output. So these random colors is just to indicate that we are able to separate cells. There's a diversity in these cell images. They can be of different modalities and different shapes and different tissue types. So our goals were to, well, first of all, to do cell segmentation. And differently from traditional segmentation, the cells are actually the same class. Usually it's just one type of cell in the image. Unlike traditional segmentation, where you may have different organs, which are different classes, or for traditional, maybe for like there's a person and a bicycle, those are different classes. And here you have the same class. And also, there are many challenges with them. They are not standardized. There are many different variations of pattern. They can be single channel, two channel in this example, multi-channel, many channel images. So we want to have the ability to work with different modalities. Of course, we wanted to make it fast on our GPUs and have all the best practices and have multi-GPU and multi-node distributed support for training. So in a nutshell, I'm going to describe how this, so this is all open source, by the way. But inside of it, your input is an image. And once again, there's a variety of these microscopy images. If you think you have, as my colleague mentioned, there's a variety of MRI images, modalities. I feel like here there's even more. There are different tissue types, there's different shapes of the cells. There are many different things. So the model we're using is based on a vision image transformer. And primarily we use VAT from a segment anything. Here's a reference. So we extracted this part and use its own pre-trained weights as a baseline to start from. The segment anything model by Meta AI, it was trained on a millions of images. They were not necessarily medical images or cell images, but nevertheless, there is an advantage to use the pre-trained backbone to start with, especially since we don't really have a lot of these cell images that have ground truth labels. We use a simple decoder, we train from scratch, and for the output segmentation, like I said, it's unconventional problem because we can have thousands of cells. We don't know how many in the output. So here we adopt a wonderful framework from a CellPose. So CellPose itself, and here's a reference, is a great open source project too, which includes a code and a network for training cell segmentation, as well as they provide even their own public data set. So we use the insight here to output, instead of the output segmentation masks, traditionally we output these vector flows, which point from the boundary to the center of each cells. And there is a post-processing step to convert it all to final segmentation of individual cells. We like this component. In fact, the CellPose is a dependency. You need to pip install it, as well as segment anything is also a dependency of this project. So these components are being reused from these great projects. All right. Oh, and here to mention, everything else we wrote our own. We wrote the whole training loop pipeline using all the Monai best practices and using Monai building blocks, so that if you use our code as a reference, it's really easy to use it, to customize it. You will see all the familiar Monai blocks. So it's written to be modular. It's written to be as a tutorial if you want to customize the code, or you can also use it as is. One example here, I want to show it. For example, this is a screenshot from this VISTA-2D GitHub repository, and this is a chain of input transforms for training. This there is a loading image transform not shown here, but then there is a random special crop transforms, random affine transformation transform, random flip, random noise, contrast adjustment, Gaussian smoothing, histogram. There's a different simulation of these augmentation transforms. They're not part of the VISTA repository. They're all part of Monai. So we just import them. It's really easy to reuse these building blocks. And you can see that you can, for example, customize it. You can comment some of these transforms. You can add your own, maybe sophisticated augmentation transforms. The last step is it transforms to convert labels to the flows, which is just a thin wrapper around the cell pose code that does it. But again, it's easy to see that if you have your own custom code, you can just wrap it and add it as a transform. So you have a sequence. This is Monai framework standard. Yes, overall, I think we made the code readable and performant. Now, as far as the images that we used, so primarily we wanted to make it the code for you to train on your own ground truth data. Maybe you have your own proprietary medical data, but to convince ourselves it works well, we have assembled around 15,000 images from a different, mostly public data sets we were able to find, shown on the screens, including the cell pose data set, different challenge data sets, and different public data sets. TissueNet is very nice, larger data set that includes different tissue from different organs, including breast and skin and prostate. And there is a variety of different modalities, grayscale images, color images, multi-channel images. And in our GitHub repository, we actually show the exact steps to reproduce it. So you can re-download all these data sets, and there's a code to pre-process these data sets so that then you can run VISTA-2D to train directly. We also have our own pre-trained weights if you just want to use the pre-trained weights right away. For the coding part, like I said, we used all the best GPU practices. It is based on Monai, it's Monai native. We used all the Monai components. Monai itself is based on PyTorch. We have adopted many things from these two different projects. From a cell pose, we adopted the 3-channel output, which is a nice way to segment these many cells without knowing how many there will be in the output. We have used SAM, well, only its vision backbone, primarily for its pre-trained weights to boost the performance, especially if you don't have too many ground truth images for cells. We did survey the literature for the just best practices overall for training and included the best augmentation transforms we could find. Okay, so this is a screenshot from our blog post. By the way, we have a blog about it, or several from the spring. So this is something we wanted to compare. We wanted to see what is the performance of this model, something we call the generalist model, because we combined so many different data sets and they were quite a bit different and quite of different shapes of cells and organs, and number of channels are different. So we wanted to see what is the performance of this model in green versus if you train like individual specialist model dedicated, for example, to only single tissue type, let's say breast. And we found that actually overall, the single model performs better on most of the tissue types, which shows there is a power, first of all, in using more data to train it, and secondly, the backbone based on the vision transformer was powerful enough to learn all of it. All right, so like I said, VISTA-2D is an open source project. You can find it under project Monai in a VISTA repository. This is the same VISTA repository as VISTA-3D project is, as was shown by Yufan, my colleague earlier. It follows the Monai bundle format specifications, and it lives inside of Monai Zoo as well at this directory. It includes the pre-trained weights. because it follows the MONAI bundle, you can use it everywhere else where you can use MONAI bundle. For example, you can use it inside of MONAI label inference server. And one example of using it I'll show you is with the cell profiler plugin, which we also enabled. So if you're a researcher and working with microscopy images, you probably use or have heard about the cell profiler, which is a nice software with a UI from a Broad Institute, where you can visually load images and then select different image analysis operation that you want to perform on images, maybe calculate statistics or do some processing. So here we added a simple plugin to run VISTA-2D from the UI. So there is a tutorial how to do it in details, but in a nutshell, you have to run MONAI label inference server on the same computer or on a remote machine. You would load VISTA-2D bundle, and then from the cell profiler on your laptop, you would enable this plugin and you would just communicate as a client server, send images and receive segmentation results. In the UI, it would look like this. You would enable one of the steps to be VISTA-2D, and then just specify the IP address where the MONAI label is running, which can be, again, remote server or local machine. Yeah, so, but you don't have to use this UI. You can just run the Python code directly, or yeah, you can just run it directly. All right, so let's see. So in summary, so VISTA-2D is a cell segmentation training pipeline. We did put a lot of effort to make it modular and readable for you to train on your own data. Although later we realized that not too many people actually have the training data. We do have our own pre-trained weights that we train as an example. The kind of results you would expect is your input can be one channel, two channel, multi-channel input image, and output will be an index map with all the self-separated. We use all the best practices for writing the code, best GPU practices. We enable multi-GPU support. It is open source. It is of interest to the companies in the microscopy and spatial transcriptomics space. At least we've been getting a lot of interest from companies in this space, where the cell segmentation usually is not the end goal for them, but a component in a workflow in their overall analysis of these microscopy images. Could be for just for analysis for itself or for different drug discovery. So they use it as a building block. Yeah, the code base is available here. Yeah, and thank you so much.